This presentation will be on amoxicillin's toxic effect on the hepatocytes in the liver, mainly the hepatic bile ducts. Amoxicillin is a moderate-spectrum bacteriolytic beta-lactam antibiotic used to treat bacterial infections caused by susceptible organisms, acting by inhibiting the synthesis of the bacterial cell wall. It is a synthetically made compound, a white crystalline powder, and is orally ingested in pill capsule form and readily absorbed in the GI tract. Properties affecting absorption. Amoxicillin has three acidic groups from 2.71 to 9.55, an alcohol group, an amide group, and it consists of an alcohol group, an amide group. Maximum stability at 37 degrees Celsius of amoxicillin has been observed to occur at 31 degrees Celsius of amoxicillin has been observed to occur at a pH of 5.77. Amoxicillin overall has relatively high polar, as well as non-polar benzenes and a four-membered nitrogen benzene, and a four-membered group shown to the left here and highlighted. An amide group, an amine group. Amoxicillin has a log p-value of 0.75, putting in the drug-like boundary range. This allows it to diffuse readily into most body tissues and fluids, with the exception of brain and spinal fluid. The half-life of amoxicillin is 61.3 minutes. Other properties. Lactic molecules such as amoxicillin are generally susceptible to acid and base catalyzed hydrolysis of their cyclic amide. To avoid this, the drug is always administered with clavulanic acid, a lactamase enzyme inhibitor. Absorption. Amoxicillin is stable in the presence of gastric acid and is readily absorbed after oral administration. Amoxicillin is well absorbed at different rate and extent from various regions of the gut. It enjoys widespread clinical use not only because of its broad antibacterial spectrum, but also because of its high oral antibacterial spectrum, but also because of its high orals occurring within 1 to 2 hours. It's widely distributed to many tissues including the liver, lungs, prostate, muscle, bile, and various other places in the body. Amoxicillin can diffuse travel through the membrane by one or two routes. First, it's relatively small molecule can diffuse through the cell membrane of the GI tract. So these two factors, because of its hydrophobic nature, relatively small, as well as easily be passively diffused through membranes. The PEP-T1 transporter contributes to the absorption properties of metabolism and is responsible for the absorption of the majority of the body's amino acids. The absorption kinetics of amoxicillin follows a zero-order model despite its slightly ionic character in the intestinal pH. The presence of this transporter helps to account for the excellent absorption of amoxicillin. Therefore, two factors contribute to the absorption of amoxicillin in the GI tract: passive diffusion, because of its hydrophobic nature and relatively small size, and active transport by PEP-T1 transporter. The way amoxicillin is actually transported by the PEP-T1 transporter is by its attachment by way of linker, if necessary, to a hydrolysis-resistant PEP-T1 substrate carrier. The entire prodrug is then recognized as a PEP-T1 substrate due to the presence of the carrier and is transported from the intestine by the transporter. Metabolism in the intestinal cells, bloodstream, or liver will eventually release the active drug. The metabolism of amoxicillin is only about 10%, only about 7 to 20% of the active. So a lot of the papers I have been reviewing general mechanism for the breakdown of beta-lactams, which is amoxicillin, is with the beta-lactamase enzyme paired with water along with CYR20, an antibody. First, a phase 1 acylation is performed to start the mechanism. First, the hydroxyl group is activated, and then the CYR70 performs a nucleophilic attack on the carbonyl group of the beta-lactam, or in this case, amoxicillin. This results in a high-energy acylation intermediate. Protonation of the beta-lactam nitrogen leads to the cleavage of the CN bond and the formation of a covalent acyl enzyme, which adopts a lower energy state. Attack by catalytic water leads to a high-energy deacylation intermediate with subsequent hydrolysis of the bond between the beta-lactam carbonyl of CYR20. Deacylation regenerates the active enzyme and releases the inactive beta-lactam. So as you can see here, the beta-lactam is actually inactivated, and you can see that most of the original structures that started the mechanism are recreated again. Excretion of amoxicillin is predominantly renal. 70% of the administered dose is unchanged and recoverable in the urine via kidney excretion, leading to very high urinary concentrations. The other 10%, because 77 to 20% actually bound to human plasma, primarily the protein, we need to account for the other 20% albumin, which 10% of it is actually metabolized. So the other 10%, now how does it get there? Is excreted. The main target pathway in terms of amoxicillin toxicity is the route from the GI tract to the liver. A small percentage of amoxicillin travels. From the stomach and is first excreted via mucosa through the epithelial cells, and then mucosa through. It then travels to the hepatic portal vein, then into the liver via the hepatocytes. This is one of the two sites where the toxin can induce hepatocytes, the inflammation of the liver, and is characterized by the presence of inflammatory cells in the tissue of the organ. Second site of toxicity is the hepatic bile ducts. The mechanism of toxicity of amoxicillin is relatively unknown. Generally, they're not known. Most papers give a general guess, and it's also to be noted that the parent toxin of amoxicillin is actually amoxicillin itself, because only 10% of it is actually metabolized. Parents generally just excreted right away. The only data relating to amoxicillin toxicity, HLA, is to bind to the antigens, or also known as HLA class 2. The immune system uses the HLAs to differentiate cell cells from non-cell cells. Any cell displaying that person's HLA type belongs to that person and therefore is not an invader. The way amoxicillin works is that it directly binds to the HLA molecule or protein antigen, and there's a conformation change effect. So the HLA changes its structure. This triggers a hypersensitivity autoimmune response. No papers have shown the exact mechanism of amoxicillin binding to the HLA class 2 antigens. HLA class 2 molecules are required for antigen presentation to CD4 positive T cells. So these activate pretty much the autoimmune response and then the cell's attack. Because of this conformational change, the immune system no longer sees the cells as self and are attacked. There are two results after the autoimmune response brought on by the change in structure of the HLA. VBDS, which is the cells are in the hepatic bile duct. Vanishing bile duct syndrome is the result. So pretty much amoxicillin binds to the HLA in the hepatocytes, or the hepatocytes in the hepatic bile duct, and they get attacked by the immune system. There will eventually be a loss of the exact mechanism of this in the hepatic bile duct. And you can see here the black arrows cut, and you can see the whole general guess of what happened. But in this picture, you can see the holes in the liver brought on by this autoimmune response. If VBDS gets worse and worse, if VBDS gets worse, cholestasis, then it has an condition where bile cannot flow from the liver to the duodenum. If the bile ducts undergo significant damage, like in VBDS, the bile cannot flow, forming a plug. This causes a sufficient amount of pressure, so the bile accumulates, and the cell ruptures, spilling the bile in the surrounding tissue, causing hepatic necrosis. Amoxicillin is generally used as a therapeutic for antibacterial infections. Seen here is there's a general mechanism at which it stops these bacterial infections. Amoxicillin binds to penicillin binding protein 1A, located inside the bacterial cell wall. Amoxicillin binding to penicillin acylate to the penicillin sensitive located inside the bacterial cell wall. C-terminal domain. By penicillin acylate the penicillin sensitive inactivation of the enzyme prevents the formation of a cross-link. This inactivation of the enzyme prevents the formation of a cross-link of two linear strands, peptidoglycan strands, inhibiting the third and last stage of bacterial cell wall stages. Bacterial cell lysis is then mediated by bacterial cell wall autolytic enzymes such as autolysins. It is possible that amoxicillin interferes with an autolysin inhibitor, but it's not well known or confirmed. In summary, the exact toxic mechanism of amoxicillins aren't known. They generally know what happens after a prolonged dose of amoxicillin, like liver damage, like VBDS, cholestasis, among a few other things. But the exact mechanism of how amoxicillin binds to HLA or inhibits transfer proteins is not known. The parent toxin of amoxicillin is its original structure. The main target of amoxicillin is the liver in terms of toxicity, and and the hepatic bile ducts. Nearly 90% of the oral dose is unchanged and excreted renally. Metabolism doesn't greatly affect excretion or the mechanism of toxicity. I mean, it's 10%, and even then, it just turns into an acid, and then it's excreted. And that's pretty much it. Thanks for listening to this video.